interested in the growth hormone. So most of you have seen by now pictures of growth hormone, spiky looking thing. It produces finger-like processes called philopodia, which interrogate the environment, pick up guidance cues perhaps, and also these flat sheets of membrane extensions which are called lamellipodia. And by picking up attractive cues, let's say maybe on this side, this whole system will turn and move towards them. So one of the things got us interested here is an observation that a couple of labs made and we ourselves did is if we reduce the amount, so if we did a knockdown complex called R23 complex, we still found this to be largely intact. Produced philopodia, lamellipodia extensions, and why we thought this was interesting is because these processes, so what makes these philopodia and lamellipodia is the cytoarchitecture of the cytoskeleton and largely actin inside it. So philopodia would be made of long bundles of actin. So if you imagine there was a rubber sheet in front of me, if you had this arm and you push it through, what would come out on the other end is a kind of a pointy structure. Right? Whereas lamellipodia would be more like if I took my hands like this and kind of pushed the membrane here, right? So you have a flat sheet, right? So one of the key aspects of regulating actin dynamics is regulating its nucleation. And traditionally, this is done by R23, very commonly in non-human cells. So we found it surprising that a couple of other people and us in our system. So what I will talk about is actually largely using developing chick as a model organism, the developing chick embryo as a model organism. So in this system also, it seemed to produce philopodia quite fine. So we started looking at, and there was one little inspiration which came from some indirect work. Yeah.
Other people have found similar results. Is it in the chick itself or other?
No, in other organisms. So, mouse, both of them were in mouse. The other inspiration for looking at this was when I was a postdoc, I was interested in a protein which many of you are indirectly interested in, I guess. This is the large receptor tyrosine phosphatase. And while the rest of the lab worked on aspects of lipids and other things, I had tried to do a mini proteomic screen because large is also guidance receptor. And to see what are the things it interacts with. And the biochemical complex that came down actually had a couple of formins in that. So what we thought, so what are formins? So formins are basically a relatively newly discovered class. I mean, it's not that new anymore. These are also known to be actin nucleators, right? So we thought, why don't we look at these actin nucleators and see what they do? And what we have done is looked at where they are expressed. Some of the work more recently, as you remember, Key was here. So we found that some of these actin nucleators are found in developing nervous system. So maybe I will stick to one because there are some variations in how they work. So let's take one example where we have probably the most information. This is called formin 2. This was found by Sid Wheeler's group quite some time ago. And it's expressed largely in the system in mouse, in the developing nervous system and adult nervous system, and in the germ cells. And most of the work that actually with formin 2 has focused on the germ cells and its role in meiosis and things like that. Mutations in formin 2 have been found commonly present with women presenting with infertility problems, etc. There's a meiosis defect. So what we found was at early stages of development with the spinal cord of chick, we find this formin expressed. And also in chick, it is enriched in the developing forebrain and the spinal cord. Right? So I'm going to say these things fairly quickly, so stop me if you can't make the connect. So the expression at the right place, the levels at the right place. One of the things, so the obvious thing to do is try and knock down this thing and see if there's any defect in axon guidance or outgrowth in vivo. So we have a system where we take an egg, fertilized egg, and we make a window in the egg. So you can see the developing embryo somewhere here. And the developing embryo, if you were to look at it from the top, there's the neural tube, which is still not closed. You can inject into the neural tube anything you want to introduce into this. You could put in GFP tag RNA, you could put in siRNA, you could put in morpholinos. That has been our method of knocking down genes. And what you do is you then electroporate this. So this is all happening in the you electroporate this, and the DNA will move into this side, right? The neural tissue going near the positive side. So what you have in the system, and then you let this embryo develop. So we basically put a sellotape on top of it and let it develop for a few days. Then you can take it out, and then you fix the embryo, stain, image, whatever, what have you, and you can assess the pathway, the neural trajectories, and see if there are any defects. The interesting thing, of course, is that one half of the embryo receives your whatever treatment you're doing. The other half is an inbuilt life control, right? So what we found was, one of the clearest defects we found was, so this is now, let's say, cross-section of the spinal cord, right? So this embryo would be about, at the stage we do is around stage 14, so this is going to be a centimeter long, right? So this is fairly big, and we let it grow easily till stage 26, it's a little bit bigger ones. Yeah.
Can you just elaborate on the fact that one is not getting and otherwise is getting?
Yeah, okay. So if I now look at this as cross-section, where I had injected was the neural tube. Right? I'm looking at this developing neural tube in cross-section now. And I have, what I am placing here is 2 electrodes on 2 sides. Right? Your nucleic acids are negatively charged. So they're going to move this side where the positive charge is. So this part of your tissue will receive whatever you are introducing. This one will won't. Right? So within the system, one side, so it's an unilateral electroporation. Right? So you have to do all these controls. I mean, when we first started doing this, we spent a lot of time even optimizing the right voltage to apply so that it doesn't kill. All of these have controls with control reagents, morpholinos, which are checked for normal development till the stage at least we want. Only then you will take it for further assessment. Yes. So those would be all of the things.
Where do you put the controls? Outside the shell?
No, just on. So what is happening here? These are where the somites are forming, right? Next to the neural tube. So you put it on the somites, just outside or 2 sides of the lateral side of the neural tube. And we also choose kind of a rostrocaudal axis so that we assess always at the same level, because there's a gradient of development from rostrocaudal leaves. So we would electroporate always between the fore and the limb hindparts, fore and the hind limb parts, and then when we match with controls, we make sure we take the same sections in the right place, right? So now in this cross section, we will use a different color here. One set of interesting neurons, and there was a reason why we were interested in it. We had looked earlier at the Drosophila homologue of this forehead, and what we found was a guidance defect very specific to the midline of Drosophila. So these are commissural neurons which are on one side of the body. They cross through the midline to the other side, and the guidance paradigm is very well characterized. It's attracted by nectarine, then repulsed by slit, so on and so forth, right? So we had seen very specific effects in that guidance in Drosophila, which is very different, of course. So one of the pathways we paid particular attention to when we looked at the vertebrate system would be an equivalent system. And the equivalent system is there are commissural neurons whose cell bodies here. They project to the floor plate, largely attracted by nectarine, sonic hedgehog, and other things. And then once they reach the floor plate, they are now repelled by the floor plate, again by slit-floor interactions. They cross to the other side and move into the plane of that board, right? So among other things, we looked at these things. And what we found was at the stage we are looking at, stage 26, the normal commissural neurons start here, go here, and they've already crossed and are well on their way. Whereas now when we use forward 2, these guys still, many of them don't reach the midline. So they're a little slow in getting to where they should have been. Right? So this was at one level kind of satisfying. We thought, okay, it's an active nucleator, maybe it's not producing philopodia and all of this, maybe this guy is just moving slowly, right? So we now decided to look at these guys in vitro in culture. So we isolate the commissural neurons, which is, it's really not an isolation, it's enrichment. So what we do is so there was one more thing. If you now take this thing, this cross section, this is a tube, you can cut this part, just this top part. So it's like this, you cut the top part, you open it up like a book, right? And it's called an open book preparation. And if you look at this in this format, it looks, one half looks something like this, and the other half looks something like this. So the spine of the book is basically your floor. You opened it like this. And if you were to label only neurons on one side, you would see they go like this, cross the midline, and most of them take a very sigmoid trajectory. So they kind of take a turn, which goes up and takes a turn and goes through, right? So we have a way of labeling this, and one of the things we see, and this we need to confirm with more experiments, is that instead of taking this very typical sigmoidal turn, there's a significant proportion of neurons which either go straight or kind of go like this. So there seems to be guidance errors that are happening. Right? I have a feeling this is distinct from this thing, which is they seem to be moving slower. Maybe they are late in arriving, the guidance cues have slightly changed, so they can't find their way, or there could be a completely distinct effect that forming 2 does. Right? But we haven't addressed this anymore beyond this right now. But I'll tell you a little bit more about this guy. So maybe I shouldn't have typed it. So again, maybe let me just redraw this. This is the open book preparation. So now you can already see if the cell bodies were here in the open book scenario, these commissural neural cell bodies would be somewhere here, right? When you open this thing. And motor neurons would occupy a different place, so on and so forth. So we open up the neural tube and we can isolate just this part and grow it in culture, right? Primary cultures. And what we see is now the idea would be if we are growing slow, you would expect these guys to grow slower than their control morpholino treated control. That doesn't seem to happen, or even if it happens, it's fairly weak, right? Yeah.
The signaling, that is the brain signaling or result signaling, it's already.
It is, as far as we can tell. We haven't specifically looked, but several other people use the same system with electroporating other things. So we haven't specifically looked if it is, but the electroporated side which didn't receive the nucleic acid reagents that we're putting in seem to be okay, perfectly okay. So, that's one advantage of doing a unilateral electroporation.
And do you see any interaction with the motor neurons which will be towards the...
Yeah, so we think we have a motor neuron defect as well, but we haven't done anything more about it yet. So they come out as a branch and kind of spread out. We think there are defects in this initial bundling. They can't seem to come out, they're a little bit more spread out, the axons, right? Again, we have not done anything more with that. However, we compare morpholino based knockdown, forming 2, and the control. They seem to be growing at pretty much the same rates, which was kind of disappointing. Till we did an experiment where we did the same experiment, except instead of using your regular tissue culture media, we take media that is, we would dissect out that floor plate region. So this was like this was the floor plate, and grow it in culture and use that conditioned media, right? And then we start seeing some things. Now you see that these guys are probably going to slightly slow, which means forming true activity is important signal downstream of something that is coming out from the floor plate. We still have to see what it is. It could be netrin, it could be sonic, it could be GMF, we don't know. Right? So the other thing that was, so what did we think it would, we started off saying, active nucleators, philopodia, maybe someone else does it, is it forming 2? However, when you look at these guys in culture, the philopodia seem to be largely okay. They're about the same length, same number, their dynamicity is fairly similar, so there must be something else. So one clue that we have is that these guys may be actually doing something slightly different. Philopodia are okay, but in order to move, you need to do a few other things. Right? So if you, in order to move forward, you throw your leg out. Let's say that's the equivalent of your philopodial or lamellipodial protrusion. You make an attachment at your feet and use forces generated by your musculoskeletal system as traction to move forward. Right? So the idea is now, can we measure traction forces and see if that is compromised, right? Philopodia saying okay, right? So the way we have gone about measuring traction forces, I think Sampada is about to follow up on some of this work, is use something called our version of what is fancifully called a traction force microscopy. So what that involves, so normally we grow in your primary neuronal cultures, any cell cultures on cover slips, right? Glass cover slips, which are infinitely hard relative to the kind of magnitude of forces cells produce. But if you were to grow your cells on top of a gel, let's say this is a cross section, this is your cells here, on top of a gel which is not as rigid, or it's flexible enough that the cell pulls on it, so it makes focal contacts. That's where the traction forces are generated. You can think of the focal contacts or focal adhesions, as they're often called in non-neuronal systems. That's where your feet is, or at the feet of the cell. Right? So if these guys pull in order to move, they will also pull the gel. Right? Now if you have beads on this gel, right, and you image the beads, these are fluorescent beads. If there's a pull here, this bead will now be pulled in a particular direction of the pull, right? So now if you were to take images of these beads, with and without the cell moving, you should be able to see how much these beads are displaced, right? And you can get a traction force. In fact, you can make it extremely quantitative, because basically Hooke's law, force is going to be proportional to the displacement. You need this modulus here. That can be calculated by finding the Young's modulus of the chain. So traditionally, put a steel ball, see how much it depresses the system, take it out with the magnet, and see. And you can find it out that way. We have also used AHM to basically find out what the stiffness is. But for most of the things I'm going to say, you really, even without this, you can get a reasonable answer. So one of the things that might be happening here is that, so we also, before going into what we think is happening, we look, let's go back to the growth hormone one more time. So this is my terrible drawing of the growth hormone. But we looked at the focus, so we use total internal reflection microscopy just to see things that are happening very close to the surface. And one of the things that happen very close to the surface is these focal adhesions, right? So we looked at focal adhesion markers, and we see that there are focal adhesions at tips of philopodia. There are also focal adhesions in the central region of the growth cone, right? And those interested, I have some of these movies, they're not in PowerPoint, but I can show you these pictures. There are certain components like zixin, yeah.
So the force element, so I think
okay, yeah. So can I go to this paper?
Where is the key?
Okay. Okay. So let's say this is a piece of paper, and this is my finger, my palm is the growth cone, right? If this is flexible, right, when this moves, I'm going to crumple it, right?
So tips are
yeah, you're doing everything except you're growing the cells instead of on cover slip on a gel which is deformed, right? So now, let's say this writing here, it was originally at this position. When there is some force being pulled on it, it will move, right? So this displacement that you're seeing is directly proportional to the force, right? So that's all I can give you. We don't do this on collagen because it's not optically good. We actually do it on we do it on a polyacrylamide gel simply because when you run Westerns, you can change the concentration of this and acrylamide, you can get different stiffnesses, right? People have done it on flexible PDMS substrates and multiple host of other things. Right? So these focal adhesions, if you look by curves, there is a subset which stays in the central regions, and there is a subset of focal adhesions at the tips of the body. Now there are some proteins that will only go to these guys. For example, zixin only goes to these guys. There are other proteins that will only go to the tips. You won't see them in the focal adhesion in the central region. Right? Also, what you will see is these guys, if you were to follow them on a time lapse, firstly they are bigger in size. Their shape is typically ellipsoid, and the long axis is oriented towards the center, right? Even in a static picture. But if you follow this over time, you see these guys appear to be as they are being pulled centripetally towards the center, right? Whereas these guys don't show that appreciably, right? So now when we look at some of the force maps that we get up to fraction force microscopy, we see that the magnitude of forces exhibited by these guys here, right, are fairly small, whereas these guys exert much stronger forces, holding force. Now, we do it in a little bit more detailed way.
How do you isolate the different forces?
No, so this guy is growing on polyacrylamide, which has beads all over.
Yeah.
Right?
So the cell pulls on it.
So when beads next to the central region is being pulled, the contribution is from this guy. Right? When a bead here is displaced next to the filopodia tip, the contribution comes from the focal adhesion there. Right? So there's a better way to do this, more direct way. What we should be doing, we haven't done this yet, is you put in GFP label tags for these guys, and you only assess the beads that are adjacent to this. Right? We haven't done that. We need to do it. But this gives you a general idea of how this, and this is not something we've been doing for a long time. Yes, and it's a huge challenge being an Sampada and how bad it can get sometimes. We have to come up with ways to make sure they are distributed uniformly, not clumped, yet enough individual beads with enough density so that they're single, so that we can measure this. So all of it is. I think we are there now, finally. So, right?
From this axis, technically you are finding out which region of the growth hormone is exerting more force and which is exerting less force.
Yes.
Not very good at adhesion proteins.
No, we have not looked at that. So one thing we've done is we've tested in the following two knockdown whether adhesivity is disrupted. Right? It doesn't adhere. That doesn't seem to be the case, though our assay is fairly crude. What Abhishek has done is he has taken these guys, plated them, and then after 6 time intervals, washed things off, and then you can go back and count how many are adhered. Right? These guys seem to be comparable as the control morphology treatment. Right?
So, how do you quantify this? Like, do you take a region, a radius around the?
Okay, so we have kind of a homemade, not very high throughput way of doing this, but basically it's an image detection system which picks up the beats, it matches it with successive frames and looks at the displacement. We have to do other things because there's always going to be a drift in the system. We have to kind of fix
Even
it.
if there's no drift and the cell is pulling on the gel, you get a displacement across a large area, and even the points which are not exerting
Not immediately close by, you get. So what we map for here is a set of arrows, right, for this. We cannot tell if this is the only one that's doing, but it looks like the closest ones are the highest magnitude and the tallest. Right? Again, so if you make the system too soft, then this effect spreads. So you have to spend a lot of time optimizing what seems to be good for your system. And the drawback of all of this is not a new technique. Right? This is nothing fancy about it. People have been doing it for a long time. But largely for non-neural cells who exert actually a lot more traction forces than these guys. People have done it for neurons as well. A couple of people have done it. So what we see is that first there's this difference, right? And our feeling is what happens is that the knockdown for M2 it generates less forces in primary tooth. So, few ways we can look into this is one, these focal additions, if you do a time lapse from the start of a focal addition forming, and maybe a couple of minutes when it matures. What happens particularly in the central ones is they start off kind of like this, and let's say this is the center, so they get pulled inwards and they finally mature into a bigger, more oval-shaped focal addition. Right? And this has been characterized in other systems as frictional slippage-based maturation. So you can measure the centroid to centroid distance here, right? And you can also do experiments where you blow myosin and you see this reduces considerably because this process is supposed to be myosin powered. But it is possible that this kind of assay will give us a more quantitative feel for what form to is. So we would expect from what our previous data suggests that it is basically going to show something compromised here, right? But the other thing we have looked at is what is called at the tips of the filopodia. So these guys go out and attach, right? And they normally don't just go and grab onto something and pull, right? They undergo cycles of load-fail cycles, basically. They attach, pull, sometimes they'll slip, they'll grab and pull. So you can measure the displacement of these here, and it looks something like this, right? And one attempt that we had made suggested that if you now look at these kind of analysis for where you have knocked down these bodies, or what you see is that they also undergo load-fail cycles, not that they don't, but if you average the magnitude of the force that is being generated, that appears to be compromised, right?
What will the addition, what is the addition of the
Okay, maybe let me finish one other thought and maybe I can try and answer that question because there is an importance to that. That's what I was going to go and say next.
In this quantification, why do you need to average it? Why don't you just ignore the negative deviations? Like when the force is being exerted.
Okay, so our measurement is there's no negative in this case. There's zero, no displacement and displacement, right, in one direction. So now there will be some which are higher occasionally. These load-fail cycles aren't even with the regular. So we take it for some time and then take an average of the amplitude. Yeah. We've not done it many times, but a few times I expanded to look at it very hard. But exactly, that would have been one of the things I would like to look at. So the other thing, which is now a little bit general, is that, yeah. One of the yes, yes. We do that. We borrow something from this guy, Nika Dengo, and basically we borrow. The second question was that after whether you can figure out the places that are essentially in the plane, where you're pushing downward. Yes, we can't. We do it with biological microscopy. There is a paper now in DNS, not too long ago, last year, which has combined with some 3D rehab. There are ways of improving. There's a really nice paper which now allows you to use turf on these systems. They've found some kind of silicon with the right refractive index so that it turfs very well. So this would be another thing to try. And properties of that polymer is such that you can probably get higher density of peak distribution without clumping. But it's hideously expensive. Yeah. Why is it there? Yeah, this is my next thing. So when we were looking at it generally, forget what we're doing now, the pattern emerges. So normally what is thought of non-neuronal cells movement is they produce a filopodia, which makes attachment, grabs onto things, pulls, right? So there's strong traction forces in front, and there's also others from the side, and then there's retraction, and the system has moved forward, right? In this case, if you look at it, it throws out filopodia, right? these guys attach, right? The magnitude of the forces, if you measure, are fairly low. The strongest forces are centripetal. Right? So what we think is happening is that these guys put out philopodia in front. They are not the ones doing a lot of the pulling. But centripetally, there's a compression. And it's like a balloon. You take the base of the balloon and you compress, you're going to get a protrusion in front. You're going to conserve volume. Right? So what we think is happening is this compression here pushes the material forward. Right? And philopodia have a relatively minor role in kind of pulling itself out. It's not like hand-in-hand movement in this case, if you will, but they perhaps have also a role which we have not looked at. Kind of, when you push this thing moving forward, you also have to direct it in the right direction. Maybe that's one of the things that these guys are doing, right? So I think that's what we do all kinds of other things with behavior of zebrafish. It's unrelated, I'm not going to discuss that. But, so this is what I think one of the central things that we're doing now. But other question that came about is that, growth cone biologists, they think of the growth cone almost as a separate cell with no nucleus. It's just like far away from the mothership. And this guy kind of crawls around fairly independently, and something might be happening in the cell body, and the axon in between, it's kind of lame and passive. Right? So various sets of experiments, including some done by Pramod at one point, suggested that this was not the case. The axon even might have an active response. So there is another set of experiments that we do together with Pramod. He's already explained some of these things in his force device, so that's how we relate to Pramod's stuff. But you've already heard about those things. But it all came from my side. He had done these experiments independently. And we had done an experiment, which was a kind of sad experiment, because in a much more refined form, someone else did it and published it. So what we did was, so we took the lab I did my postdoc, so I didn't do much of it, but it was largely a Drosophila lab, but I did other things. But we also did a little bit of Drosophila. So in Drosophila, the advantage is, at least at the level of the embryo, every single motor neuron is known, the trajectory is known, which muscle will go and connect to its known. It's not as good as your worms, but not bad. So what we had done was, we knew that there's this particular muscle here. And this fellow gets innervated by a particular motor neuron and forms a synapse there. Right? So it started off with a kind of a little bit dumb idea, and then it took a complete different course. We thought, what if when the growth cone is here, it's not reached its target, we cut it off from its mothership, the cell body, right? Does it still have the machinery to go there, right? And it did, right? And you see there's a lot of literature now, which in various ways shows that this happens, at least over certain length scales and time scales. And there are also mechanisms, right? But what we also did, because we were thinking of this axon and all of this, is that effectively, when you are, if an axon is under tension, let's say, so you can think of this like it's a bit of a rubber band. It's full and stretched, right? Even when it's, let's say, here, it's going to go there and form a synapse. If I cut it here, it's almost like releasing that tension, right? So what we had done was, when we cut this, we also did it with Drosophila, which express GFP synaptotachin in these neurons, right? And this, is one of, so normally it's expressed diffusely in the axon and the growth cone. When it hits the muscle, it's going to make a synapse. There's an accumulation at the growth cone. It's almost the first sign of that the synapse is going to form here. So what we saw was, in the normal case, it went there was an accumulation of synaptotachin. Then when we cut it, this guy still goes there, but no synaptotachin accumulates. So if you were to plot, this is where the growth cone is, let's say growth cone, and this is certain things going away from the growth cone. Once it's reached, you would normally see there's a peak of synaptic inflorescence and then stay diffuse around the rest of the node. When we axotomize this guy, this never happened with kind of state something like this. Right? Of course, you're going to tell me, you idiot, it has to come from the cell body and you just remove the cell body. Right? So we did a crazy experiment where we got this cut end with the glass pipette and used a micromanipulator to pull it back. So reapply the tension artificially completely. Right? What we saw was this recovery. Right? So we thought that was fairly crazy, and about this time we were already talking with Pramod. We thought we'd try to measure all this quantitatively, all of that, wrote a grant came, but someone else's paper also came right after the grant. So now we think we have to do more cooler. They of course did it in much cooler ways. They did cut and all of that. We did it with sharpened tungsten needles. They used lasers. Cool stuff. We did have those. But I think what is that this mechanical tension in this can have a real, functional role in improving neurons, and you can think of this in many ways. But maybe I end up thinking the one way this might happen. In cultures, we often see chick neurons and all of them. They are often several hundred microns long in culture, right? And they'll be producing all these branches. But if one of these branches stabilizes, you see that many of these other branches end up retracting. Have you told them already about retraction in this one? And this is happening several hundred microns away often. And at time scales, don't make much sense. I mean, maybe suddenly nothing is going to diffuse that fast, and I don't know, motors will not suddenly come back that fast. So, one other way this kind of fast communication can be achieved is, I hope I have to do a demonstration. So, if a system, this is the growth cone, and this is the other end, is if the system is mechanically coupled. So, I tug here, you see an instantaneous displacement here, right? So, if this is, an axon, you already know the cytoarchitecture of the cytoskeleton. It seems to be a cool system where this could happen over. So, mechanical coupling could form a mode of long distance signal transduction, right? And why tension in all of this? Well, if this was like this, untense, Pramod has nice words for this. I think they call it pre-stress and all of that. But if this was like this, you pull here, nothing really transmits here. It's dissipated in between. But if you have the system under tension, this will work more efficiently, right? So this is one of the grand goals Pramod and I have, and we hope to do it for other people do other things. Okay, that's all I have to say. Thank you.
So the last announcement is that tomorrow my post-dinner talk is advanced, so there will be no post-dinner talk. But tomorrow there will be the discussion groups, and then I will talk, and then we'll go through the posters. And that's it. That's the end of the day. The rest of the program will be as scheduled.